where growth factors are often claimed to be an alternative to exosomes. So I want to get into the details of the comparison between exosomes and growth factors, what they have in common and where the differences are. And that will enable you to actually judge yourself how they compare. So growth factors are either secreted by a producer cell in a physiological environment, or they can be administered by a healthcare provider. So they constitute signaling molecules that then fuse with specific receptors of a recipient cell. Therefore, you have signal transduction to the nucleus, and that leads to activation of certain gene expression for the cell. And that, of course, results in a biological response. So growth factors are indeed very relevant and very important. Conversely, when you compare it to an exosome is filled with cargo of various kinds, and I'll get into the details of that in a little while. And then the exosome will fuse with a recipient cell, introducing all their cargo, and of course also elicit a biological response. So there are similarities between the two. The main similarity, or the main difference between the two, is that exosomes are filled with a lot of different things and introduce all their cargo into the recipient cell, while a growth factor acts only on the surface and elicits a certain biological response. So, as I said, some similarities and of course some differences. Getting back to growth factors, this is a list. I have to apologize that everything is very text heavy in this presentation, and you don't have to read all of this. I'm happy to send you the slides if you're interested in those details. But just to give you an idea, these are the 8 best studied families of growth factors. There's the FGF family that alone has 23 different kinds of growth factors. The VEGF family, IGF are very important and prominent in the field. EGF, then PDGF. This one I will mention a few times because it's currently hailed as the super growth factor, and it's very powerful indeed, but it's just one growth factor. There's the NGF family that is more relevant to neural development, the HGF and the TGF-beta family. Just to give you an idea, they all have somewhat similar tasks and with finite differences between them. As I said, these are just 8 growth factor families. If you look at all the known classes or families of growth factors, there are indeed, I think, currently 17, and I try to list them here. The EGF family. Cell proliferation, differentiation, survival, maybe not so interested in our applications. FGF family, as I said, like at least 23 have been identified so far belonging to this family. Very relevant to wound healing and tissue regeneration. We have the PDGF family. PDGF is, as I said, like one of the more prominent growth factors that everybody talks about these days. And even here, there have been several isoforms identified so far. I don't want to go into the detail of all of them. You just get the idea of the diversity of those growth classes or families. There are a lot of them have to do with growth, regeneration, proliferation, and regeneration. So they're very relevant to what we are trying to do. And as I mentioned, there are currently 17 classes or families that have been identified, all of which have a certain relevance to what we are trying to do in the field of either regenerative medicine or aesthetics and cosmetics. So growth factors, as a definition, are naturally occurring proteins that regulate cell growth, proliferation, differentiation, and also healing. And they bind to specific receptors on the cell surfaces and activate intracellular signaling pathways to stimulate biological responses. They are essential in tissue repair and regeneration, as well as immune modulation. And they are also commonly found in exosomes, of course, as well as in platelet-rich plasma, PRP. And they're very, very relevant for the field of stem cell-derived therapies. Currently, there are at least 50 known growth factors. I think that's still underestimated if you count all the isoforms that are around as well. So there are a lot of them, and I get back to this point a little bit later, how this is relevant when we talk about the comparison between exosomes and growth factors. Exosomes, their cargo consists of a diverse range of biomolecules that play critical roles in cell communication, regeneration, immune modulation, proliferation, and homeostasis. So they have a much broader application or a much broader importance when you compare them to growth factors, where this is more specific, and exosomes can accommodate and satisfy and trigger a wide range of biological processes. The major classes of biomolecules that you find in exosomes are proteins, of course. I just listed a few as examples to give you an idea: membrane proteins, as well as cytoskeletal proteins, enzymes and chaperones, and of course also exosomal markers. Their function relates to facilitating exosome biogenesis, so making exosomes to begin with, targeting and uptake by recipient cells. They mediate immune modulation and inflammation control, so very relevant to what we are doing. And they participate in wound healing, angiogenesis, and tissue repair. Then we have, as part of the functional proteins, we have growth factors and cytokines. So we have the growth factors, cytokines, they're often interchangeable, and chemokines. And there's like a whole list of them. Their functions relate to enhancement of wound healing and tissue regeneration. They can regulate immune responses and inflammation, and they promote angiogenesis as well as cell survival. Lipids, also when we talk about skin and skin rejuvenation, they're extremely relevant. There's a range of lipid classes, and their functions relates to maintenance of exosome structure and membrane integrity. That's also a topic that's very relevant to when we're talking about lyophilized exosomes, which turn out to not contain any exosomes anymore, because in the process of freeze drying, their structure gets actually destroyed. So the bilipid outer layer that contains all the cargo of an exosome gets unfortunately destroyed in the process. So everything will spill out, and what you end up is exosomal debris and maybe some residual growth factors that do some benefit, but it doesn't relate to the potency of a bioactive exosome. Other functions are fusion with target cells, which is also an extremely critical and important point. In immune responses, while growth factors can do that too, growth factors are more limited and more specific, while exosomes have the capacity to signal for a wide range of pathways that enables also the cell to get reprogrammed and transferred back or returned into a more youthful and fully functional state. They also promote tissue regeneration as well as neuroprotection. Then a very important class of biomolecules are the nucleic acids, the RNAs. So there's a variety of RNAs, and I often refer to them as the instructions, as you will. So the exosomes contain both the building blocks as well as the instructions for a cell that is either damaged or senescent or otherwise impaired to regain full functionality. And you definitely need both. If I refer back to the lyophilized exosomes, not only get the exosomes destroyed in the process, but also all the RNAs get lost in the process. Proteins and growth factors are somewhat more resilient, but all the RNAs, which provide the instructions of how to do things, inevitably get completely destroyed and lost in the process. The function of those RNAs are gene expression in the recipient cells, so they modulate and govern the gene expression. And that is relevant to cellular differentiation, proliferation, and also apoptosis, or prevention of apoptosis. Immune modulation and anti-inflammatory effects are also regulated by those RNAs. Then we have other classes as well, the metabolites, among them amino acids, some carbohydrates, energy metabolites like NAD and ATP, as well as vitamins and coenzymes. If you look around in the field and what some vendors actually are trying to sell, you will notice that oftentimes they add these kinds of things. They add amino acids and vitamins and other things as well, which should make you wonder how much trust and confidence they have in their own exosomes if they feel the need to add certain things that are inherently contained in exosomes, at least provided when you actually source them correctly and manufacture them properly. So the function of those metabolites can be energy transfer and general metabolism. They influence oxidative stress and redox balance for the cells, and they regulate immune responses and also cellular signaling. And last not least, there are some extracellular matrix components like hyaluronic acid that we all know to be important for skin, to give it a more supple and hydrated self. Collagen fragments are present in exosomes, and then also things like fibronectin, laminin, and elastin, which gives our skin the flexibility and healthy, youthful look. So function, structural support, and cell adhesion, modulating cell migration and tissue remodeling, and they influence activity in regeneration in general. When I say stem cell activity, that includes resident stem cells that are inherent to our bodies. There are quite a few around still, just not enough to typically cope with any damage or aging effects that we suffer from. So the summary for exosomes, as you probably can appreciate, they serve as potent biological messengers. They're experts in cell-cell communication, and they do that by transferring multifaceted cargo to regulate cell behavior and enhance regeneration, immune modulation, and also rejuvenation. So the balance and abundance of these cargo molecules determine the potency in clinical and aesthetic applications. So again, that's why it's important to really source your exosomes cautiously. Virtually all cells secrete exosomes, but not all exosomes are created equally. So we at Resilier, as you know, source our exosomes from Wharton's jelly MSCs. And these MSCs just finished building a new human. So they are specialized in the development of tissues and regeneration, as well as in repair, just in case in the process of development something goes awry. The table is just a summary of what I just presented. And very important here is that exosomes are known to carry over 1,700 factors. So that is, of course, a big difference to the 50 known growth factors we talked about a little bit earlier. To also summarize the difference between those two entities, if you look at the growth factor, which is depicted by the receptor and the little red triangle to the left, and if you compare that to the exosomes that just fused with a target cell and released all their cargo, you can obviously appreciate the vast difference in what kind of signaling capacity is provided by an exosome versus that of a growth factor. So when you look into the market of what's offered, you either have a powerful and bioactive exosome that contains roughly 1,700 known factors, or you have another product that contains one or several growth factors. So that is, of course, a huge difference and inevitably influences the potency of what you can.
They were easy about it. They're just going to expedite them out quickly.
But I wanted to order more.
They will do it online.
So it could come in the same shipping box.
If you could mute yourself, that would be much appreciated. Thank you. Here is a very important summary, the growth factors, claims versus truth. So I mentioned PDGF, which, as I said, is hailed as the most powerful growth factor you find anywhere. A lot of people are very excited about it, and rightfully so. So it's a very powerful growth factor that does a lot of good, but it is no comparison to the potency of bioactive exosomes by the sheer virtue of the signaling pathways it can accommodate and what it does. Also very important to know is that a growth factor is always acting what I call superficially. So it has a temporary response and elicits a temporary biological response through their signaling pathways, which is temporary and not sustained, and it doesn't fix the underlying cause of why a cell is not fully functioning anymore. So growth factor can only remedy a simple or palliative and only transitional. Only exosomes have the capacity to actually fix a cell, and they do that by the sheer virtue of providing all their cargo that includes all the building blocks as well as the instructions for a cell to actually reboot and reset itself and therefore regain full functionality and a more youthful state again. There are a few black sheep that make wild claims. One of them is a competitor who claims that certain growth factors are FDA approved as drugs, which is correct. There's a number of growth factors that are FDA approved as biological drugs for various things, hematopoiesis, wound healing, bone regeneration, and certain growth disorders. However, they are highly regulated, and their clinical applications depend on precise indications. So that means just because a growth factor might be FDA approved for particular, just use it for any other indication or condition. So there's a little bit of a disconnect here, and what they do or say is very misleading. For instance, this one competitor is mixing up 4 growth factors that are all FDA approved for a particular indication, but they reformulate it, therefore voiding any FDA approval because it becomes a new sort of drug. And then they claim that it's okay to inject them, which is absolutely false and would get you in trouble if you actually do that. Interestingly, on their website, you will find that they talk about a MOCA registration for their growth factors as a cosmetic and not about an FDA approval as a drug, which they do not have. So you have to exercise some caution, not only to the wild claims as to the potency of what these growth factors can do, but also if vendors actually make you believe that it's perfectly fine to inject it because it's an FDA approved drug, which in fact it isn't. So it's sometimes hard to navigate, and I just hope to sensitize you and have been able to sensitize you about this particular problem, where a claim is not only a falsehood per se, but it could get you actually in trouble if you use their products as they want you to do. Synthetic exosomes, very interesting topic as well. There's also one major competitor out there who claims that they invented synthetic exosomes, which is one falsehood. So synthetic exosomes have been around for a long time. They're called, this particular vendor calls them a little bit different. They call them, trademark this. There's really nothing bio about it, I must say. They are synthetic, which just means that they have been created in the lab and they are empty bubbles. So they're not exosomes per se. Their structure is somewhat analog to an exosome, which means they have a bilipid outer layer. That's the only similarity they have with an exosome. And then they fill them with, I think, 10 different growth factors. All of these growth factors, again, they are relevant, they are interesting, and they are beneficial. However, they do not compare to the potency of a bioactive exosome, which is just, again, what I demonstrated or explained a little earlier. And so what they claim is that their synthetic exosomes filled with 10 growth factors would recapitulate the function of biological exosomes. And that, in fact, is not only misleading, it is simply not true, and it's a false claim. That competitor is by default a growth factor company, so it's understandable that they, oversell their growth factors a little bit, but it shouldn't lead to false claims that are misleading physicians and patients alike. And last not least, lyophilized exosomes, I mentioned them before. They do not contain any intact bioactive exosomes, as we have just recently demonstrated. It's also work still in progress. We have investigated exosomes that are out there that are sold as lyophilized exosomes, and it turns out that there are no intact exosomes included anymore. As I said, freeze-drying process destroys the outer membrane, and therefore you end up with just exosomal debris. Therefore, they can also not function as exosomes. So the whole idea of why you actually utilize exosomes, enabling the cell to get reprogrammed and regain full functionality, is not possible with these kinds of products. There might be some residual growth factors left that show a temporary and then superficial benefit, as so many other growth factors, but that's about it. So that basically sums it up in a nutshell. In summary, I can only say growth factors are relevant and very important, but just by using one or several growth factors, you do see of an exosome. That is like a whole different level. And as I said, the difference between them, growth factors can elicit a biological response that is temporary and superficial or palliative. And exosomes, they are the real deal. They can actually reprogram and fix a cell. So they can address the underlying cause of what is wrong with a cell or the tissue that's being treated. I think I used that analogy before. It's a little bit like putting a moisturizer on your dry skin. For as long as you use the moisturizer, your skin looks fine and healthy and young and supple. But as soon as you leave the moisturizer away, you go back to your dry skin. So that would be the effect that you have from growth factors. If you have an exosome, the exosome can actually fix the dry skin, and therefore you don't need any moisturizer anymore. So that's basically the whole difference between the two. It's also, I like to argue, on the level of common sense. And you have to imagine that every cell has your complete human genome. And your human genome consists of roughly 20,000 genes. All these genes at all times are active to some degree, are expressed to some degree. More or less, they're upregulated and downregulated, and that makes a functional and healthy cell if everything goes well. So the idea that you would ever be able to find one factor, like it could be the most powerful growth factor in the world, the idea that you have one factor who can fix it all is just it doesn't make sense. It defies common sense and logic, and it defies biology. So any vendor who claims as much is giving you misleading information to just push a certain agenda. And that sums it up. I'm happy to take any questions you might have.
Thank you so much, Dr. Heke. If any doctor has any questions about this topic, please feel free to ask Dr. Heke, either by unmuting yourself, or you can just write the question in the chat group.
I guess it was abundantly clear, the differences.
That's good, actually. There is a question from Dr. Mahesh. Is there any issue if someone injects exosome?
Yes and no. So I can only speak for us. We have two companies. One of them sells exosomes for regenerative medicine purposes, and the other one for cosmetics and aesthetics. And the reason is that, at least here in the US, and in some countries it might be different, but in the US you cannot sell exosomes for both applications under the same umbrella. You have to strictly separate them because there are regulatory differences. That being said, it is our parent company who manufactures those exosomes according to all FDA guidelines and regulations, clinical grade. And we do not make a difference between those exosomes that are sold for regenerative medicine purposes and those that are sold for cosmetic or aesthetic purposes. So they have the same quality. They're both clinical grade, just sold by different companies. That being said, if you buy Resilier exosomes, they are topically only. So technically you are not allowed to inject them, and we cannot encourage you to do so. If you want to inject them, you would have to resort to the regenerative version of those same exosomes.
Yeah, clear, doctor. But because these questions are asked for us many times, regardless the registration which we share with doctors, that the registration is topical microneedling, but any risk of injecting the exosome into the skin, like intradermal?
No, there wouldn't be. So as a what I say, and I have to say you cannot inject Resilier exosomes. It's just regulatory-wise not allowed. In terms of the scientific rationale or the medical limitations or considerations, yes, they are manufactured in the way that they could be injected without causing any risk or harm to the patient.
In which layer do you recommend to inject for, let's say, aesthetic purpose, like intradermal, deep? In which layer you recommend to inject if doctor decided to inject on their responsibility?
Yeah, it depends a little bit on the on the indication, whether you go for hair regrowth or skin rejuvenation. You actually do not need to go too deep because the exosomes per se have the ability to even go through the conostratum, and they penetrate as well. It is also important that you don't want to go and accidentally destroy a hair follicle, for instance, by going in too deep. And of course, you have they disperse once you inject them. So there is no really established benchmarks. There are certain things that we can recommend or suggest based on experience or what we have seen has been done and successfully applied by other providers. And if there are more specific indications that you're interested in, then please ping me and I can check with our
على البشرة كلها. الحساسية مرة في اليوم تحسب فيتامين د. يبقى ده بيبر تبدو.
What she knows.
I just muted the people who made this noise. So thank you, Dr. Heke, for the answer. There is another question. Are we ever going to see shelf-stable exosome non-lyophilized?
Very good question. As a matter of fact, we are still in the process of testing it. And it came a little bit to our surprise as well, just how stable our exosomes are, at least. We have seen them stable in a refrigerator for over a year. And we have also tested them at room temperature, and we know them to be stable at room temperature for at least 30 days. And we continue the testing. It is, at this point in time, nothing we would recommend because storage is always different from usage. As a storage means, we would still advise to actually keep them frozen until they are used. But I'm confident that yes, we will see some room temperature shelf stable products coming out of our pipeline at some point, and the skincare products enable us also to play a little bit around with the formulations to further support that stability. So the short answer is yes, not right away, but it is on the horizon.
Great. One more question. How do you make sure that exosome product is consistent from batch to batch?
Also a very relevant question. So this is actually determined by some of the FDA regulations that we follow to the T. You need to establish SOPs, standard operating procedures. That means that every time you do manufacture or you do treat a cord and harvest those MSCs, everything has to be done exactly the same way, down to the T, like with no exceptions. So even like in what hand you hold your pipette, etc. So the repetitive nature of these protocols and the strict adherence to that give you some control over it. And then it depends, of course, on third-party testing, which is also stratified and normalized. We only process cords when the cells that we harvest from them satisfy certain expectations as to their growth potential and proliferation capacity that's being extensively tested. And only when you get the green light for the cord to be further processed, we grow the cells. These cells secrete the exosomes, and then we do some internal testing, characterization, and validation. And then we have third-party testing that's more unbiased to confirm our findings. Last not least, we have trans-electron microscopy and nanoparticle readings being done on our final product. And we are currently also doing some proteomics and lipidomics and some RNA profiling for all our products. So that assures that they are as consistent as they can be.
Clear. There is another one also. She said some papers said exosomes can promote cancer, and some said exosome can help with cancer, like help to treat cancer. What is your opinion about this?
The first one I would still consider a scare. The second one is certainly true. So we have seen exosomes being applied in mediating cancers successfully. That's still experimental, and it's nothing that would be FDA approved or otherwise approved. It's work in progress, but there's a strong indication that this is a promising means to mediate cancers, to some degree at least. The concern that it could actually cause or promote cancer is usually derived from what we've seen in the field of pluripotent stem cells when they had been applied or transplanted into animals. And embryonic stem cells, for instance, have shown to cause teratomas, which are benign tumors in animals when transplanted. That is a commonly known problem. That's why embryonic stem cells are no longer relevant or considered for transplantation or infusion because they are hard to be controlled. It's too risky. But that doesn't hold true for mesenchymal stromal stem cells. So they have never been shown to actually cause teratomas like this. And exosomes derived from them are even safer because they are no living entities. They do not contain any DNA, so they do not contain anything mutagenic that could cause cancers. And the fact that there's a hypothetical concern about growth factors promoting cancers is really just that. It's hypothetical. There is no reason to assume at this point that this is really a fact. I think it is also a little bit triggered or perpetuated by the pharma industry that is a little bit, anxious or even scared of stem cell and exosome therapy, because that would have the potential to actually cure diseases rather than just treating them, which is the primary interest of big pharma. And I don't say that lightly because I used to work for big pharma. I started my career there. I want to hold it too much against them, but the pharma industry has a very hard burden whenever they want to establish a drug that typically goes in hand with 10-15 years of research and development and clinical trials. It's extremely expensive, and a lot of those clinical trials fail and very late, so it's very expensive, these failures as well. So I don't hold a grudge against them when they argue the same should apply to exosomes and stem cells. So they just caution it. but there's no proof at this point, and there's even a paper that I can send around that clearly demonstrated that exosomes cannot promote cancers.
Yes, please, Dr. Heke, if you can share with us this paper so we can share it with the doctors, as this question also frequently we are asked from doctors. One more question, how do they apply after microneedling? Is there some type of stack associated with this? And is the recommended 5 billion per facial, like per treatment you recommend 5 billion?
Of our exosomes, yes, it's a sufficient amount. So we did an extensive clinical trial, 100-person skin rejuvenation study, where we actually compared 5 billion with 50 billion and with 100 billion exosomes. And the result basically has shown that more is better, but if you use 50 billion, it doesn't give you 10 times as much of a benefit as 5 billion. Conversely, we realized that there is somewhat of a saturation. So if you do a microneedling of your face, 5 billion is typically enough. For some time, we have promoted 10 billion as the sweet spot, and you can still go for it, but 5 billion would be sufficient. And it's probably recommended to rather repeat these treatments at 5 billion each, rather than one treatment at 50 billion.
Clear, thank you. And regarding the way of the application, the question, I think she mean that do you recommend like any type of microneedling movement on the skin, like stacking or movement around the skin? Is there any difference when you do this?
I don't think so. So oftentimes what we want to achieve is pinpoint bleeding, but it has been shown that not even pinpoint bleeding is necessary. So whatever your routine is in microneedling would be sufficient to support an even deeper penetration of the exosomes, and the combination of both typically works extremely well.
Right. All right. One more question from Dr. Muhammad. What are thoughts on a recent PDGF?
Oh, yeah, that's one of the growth factors. Very relevant, and it's a great growth factor. But as a reminder, it's just one. So you cannot compare it to the potency of exosomes that contain all the growth factors typically, as well as the RNAs, which are just as important.
I see. All right, the next question from Dr. Mu'taz. What about using the exosomes carried by liposomes? And what is the role of liposomes in regenerative medicine and tissue reengineering?
So liposomes, I assume synthetic liposomes, which are very popular as vehicles to actually transfer or transport certain remedies, agents, or drugs even. Highly relevant for the field of regenerative medicine when it comes to pharmaceutical. And liposomes were created based on what exosomes really do. They can be considered liposomes as well. So they are somewhat analogs, but again, I want to caution about liposomes or niosomes that are a synthetic means to transport select agents that can be great and beneficial, but they do not recapitulate the potency that biological and bioactive exosomes constitute.
All right, thank you for the answer. The next question, how do your exosome compare autologous exosomes in high quality PRP system?
High quality RP system?
PRP system.
Oh, PRP system. Also a very good question. I'm always a fan of...
I think she's referring to something new came recently called MetaCell, which they take from the patient blood and they put it inside the device to generate exosome as per their claim.
Precisely. So PRP, we know it works. It is relatively restricted. So PRP, for instance, contains 5 to 20 growth factors. Again, compared to all 50 that you would find in exosomes, there's a difference. Autologous exosomes, that is something I'm rather wary about, and I would caution. It sounds good. I'm a big fan of clinical translation of stem cell research into medical applications. We established the patient's own bone marrow as the primary source and a good source for stem cells to treat them for various diseases. And in my case, it was heart failure. And we had great success. It comes with a downside that you rely on whatever number of stem cells the patient has, or whatever quality they have. And here is the big problem that I have with it. If you have autologous exosomes that are derived from the patient's peripheral blood, they are not characterized, they are not validated. You do not know what these exosomes are. You do not know anything about the quality of those exosomes, how potent they really are. A patient might suffer from comorbidities like age-related issues. They might even have certain mutations accrued throughout their lifetime already, all of which would have an impact on the quality and the potency of the exosome. So that's a complete black box, and the only thing that seems to speak in favor of autologous exosomes is that it's from the patient and going back to the patient. So patients sometimes feel very comfortable about this rather than having an allogenic option where it comes from somewhere else. So they feel more in control, but the quality and the potency are a far cry from what they can be if you derive your exosomes from Wharton's jelly, for instance.
Okay. How often should exosomes be used?
Again, it depends on a little bit on the indication, the severity of the indication, your age, etc. But what we have seen applied quite successfully is usually to address a given indication. You have 3 stints at the beginning, let's say with 4 to 6 weeks in between, and then you enter maintenance where you actually do it once, twice, 3 times a year. And again, it depends a little bit on the personal response and the severity of the indication and how well it was addressed in those treatments. And then you can modify accordingly by either increasing or decreasing frequency and the number of exosomes applied. But that gives you an idea. I would always say like 3 in short order and then twice a year or so. That should, in most cases, set you up quite nicely.
Is there any limitation of repeating the sessions?
No, not really. When you do, for instance, hair restoration, you should wait a certain time because there's a natural turnover that you would have to wait for a little bit to actually see the effect. So doing it like every week is probably a little bit of an overkill, but there is no risk to it, and there is it's probably a cost factor rather than anything else. You cannot do any harm, you cannot do anything wrong by it. It might not be necessary or a little bit redundant if the frequency is too high, but it's possible.
All right. A question from Dr. Mahesh. It's hard to do Resilien exosome with Dermapen as it's suspended in saline solution. Mm-hmm. Okay to mix with hyaluronic during procedure?
You could, yeah. There's nothing that speaks against it. If you want to have it in a little bit higher viscosity, then you could do that, yes.
All right. The next, do exosome help with superficial wound healing?
Yes. Again, for the topical exosomes, we are not allowed to talk of healing, but it can improve the appearance of a wound. So it can also improve the appearance of scar tissue following a wound. So it's very effective. Yes, we have seen our exosomes also be applied after more procedures where cancerous lesions have occurred in patients, and we've seen remarkable success in applying the regenerative version of our exosomes on those deep wounds, and they close up much better than.
You mean in your case, if you are saying it's not for topical, if it's wound healing, it should be topical, right?
Yeah, it's a tricky thing. Sometimes it's semantics, that's why I'm always have to be a little bit careful. It's semantics. For instance, when we talk about scar, for instance, if you say I want to treat a scar, then it's a medical application. If you say I want to improve the appearance of a scar, then it's topical and aesthetics, even if what you do is exactly the same thing. So I just have to be careful about the wording. But yes, you can apply exosomes on wound, and it will support the wound healing process.
Even here in Dubai, we have very successful trials on burn clinics here in Dubai by using Resilil exosome. The next question from Dr. Mu'taz: can exosomal content be changed during the process of isolation?
Can it what?
Can exosomal content change it during the process of isolation?
No, it doesn't happen. So the exosomes are really contained and relatively stable. So all we really do is to isolate them, purify them by various filtration processes, during which they are not treated with any chemicals or any other means that could change their cargo in any way. So in our case, we can vouch that there will be no alterations in the manufacturing process.
And I think the testing you are doing on the final product eventually after you finish the process of manufacturing is a great evidence that the exosome is functional and active.
Precisely, yeah. That's right. Also imperative, and that's something you should technically insist on from whomever vendor you choose to go with.
Right. And would using exosome for a scar revision lessen scar or build them?
I didn't quite get that, the last part.
Would using exosomes for scar revision lessen scars or build them?
Lessen scars. So we have seen that both on fresh tissue after a recent surgery, where exosomes were applied on the sutures, and we have actually done a half scar study where you see a clear difference between the half that was treated with exosomes and the other half that was not treated. So the one with exosomes healed much faster, and it was like a fine line. The other one was a little ragged and was purplish. So that was for fresh scars very effective. And we've even seen microneedling plus exosome could reverse old scars. We had patients with decades-old scars, and they could be remedied as well to a large degree. And we have also seen our exosomes being successfully used, microneedling plus exosomes, for the treatment of acne scars.
The person who write the question can elaborate about how he or she think that exosome might build the scar. Maybe she mean that the opposite can happen, like instead of reducing the scar, it's increasing and building the scar by tissue regeneration. Maybe he or she can elaborate if she want to speak.
It's very unlikely, because scar tissue is actually an overproduction of collagen. So when you have tissue damage, there will be a production of collagen that is supposed to, help the tissue to stay connected. And unfortunately, these mechanisms are a little bit too potent. So oftentimes there's too much collagen produced, really, and that actually results in that scar tissue. When you apply exosomes, the exosomes actually help the cells to function properly. So rather than overproducing collagen, it might be downplayed cells to regenerate more quickly, so that there's no need for the overshooting of collagen. So I've never heard of any case where exosome treatments would have caused a scar or increased the scar tissue. It's very unlikely and not feasible how that would even happen.
All right, clear. The second question, is there any way that freeze-dry exosome can still remain active exosomes activity? There are some labels claim that their lyophilized exosomes still have the bioactive activity of exosome. How do you actually measure this?
We did this with trans-electron microscopy. So we actually compared a number of products that are available, including those by and we have shown, or we couldn't show, a single intact exosome. That was also confirmed by the nanoparticle analysis, that also has like wild peaks all over the place, and not what you would expect, where there's one peak related to the average exosome size of 100 nanometers. So there are various indications, and at this point I have to say proofs that there are no intact exosomes in there. Obviously, a vendor will always claim everything to be just fine. If they do, just ask them for a TEM analysis to show that they actually have intact exosomes. There's also something needs to be said about one of the market leaders, or the market leader, who also sells lyophilized exosomes. They often refer to their publications. They have like 6 or 7 publications about the use of exosomes, more in medical applications. And interestingly, those publications were always based on exosomes that had not been freeze-dried when they tested them. Only what they sell you as a topical in the field of aesthetics and cosmetics, these are all lyophilized because they have a better shelf life. Unfortunately, they're also broken, so the longer shelf life doesn't give you much of a benefit.
All right. There is a question about specific brand which recently came to the market. Are lyophilized. Does the lyophilization destroy synthetic exosomes as well as other lyophilized products?
I'm actually not even sure. I mean, I cannot use any brand names. I'm aware of their niosomes and what they are doing, that they are synthetic and that they are not analogs to biological exosomes at all. There's nothing bio about them. I would be surprised if they lyophilize them, because it is known that they get destroyed. So it wouldn't make sense to actually create a synthetic exosome and then destroy it by lyophilizing it. I know that there are other products, there are hybrid exosomes, etc. They are lyophilized, and they should know better by now not to do that. But it's the nature of the game, and they are very aggressive and very elegant, I must say. And as long as they are successful in misleading customers by their wild claims, unfortunately very inaccurate, they will continue to sell these products as they are.
Right. What is the pH level of H0 TEM exosomes, and how does it impact skin penetration and treatment efficacy?
a pH level of the TEM?
pH
analysis?
level of H0 exosome.
Uh-huh.
How does it impact skin penetration and treatment efficacy?
Okay, so the pH level of our exosomes are between 5 and 7 typically. So they are in physiological saline. So they mimic the natural skin environment in terms of pH level, and that enables them to be very biocompatible with skin applications. I hope that answers the question.
Yeah. Do you think plant exosome really work for skin?
Oh, you broke up after plant exosomes.
Do you think plant exosome really work for skin?
Absolutely not. How could they? So exosomes are experts in cell-cell communication. And to actually talk to a human cell, you would actually need a human exosome. Plants have like lots of good things about them, and you have aloe. We know that it's good for the skin, but it's all very superficial. They do not talk to our cells, and they cannot fix them. So both plant-derived exosomes and animal-derived exosomes are complete nonsense. They're often portrayed as being safer because they are not derived from humans, and that would make them better somehow, but they're neither better nor are they safer. So they could be just as risky as any other product that is not properly manufactured. But most importantly, the cell-to-cell communication is not possible because they are not the same species. Just as we cannot talk to plants or animals, they cannot talk back to us, and same is true for the exosomes.
Right. Can exosome be used to treat teen acne?
We have not actually entertained a study in active acne. We are currently having a few case studies where this is being tried, and it's a promising approach because it is actually, there's a lot of inflammation going on that is typically a target for exosomes. So it should work. We know that acne scarring can be addressed, and that should be true for active acne as well as for old scars. So the answer is most likely yes, but it's still in the process of being studied more in depth.
And probably for active acne, Dr. Michael, it has to be injected. We have very good experience with active acne with Resiliel exosome with doctors here in UAE, but it was injected because we cannot use it with topical microneedling for active acne.
I agree. Like microneedling is out of the question, just as many other adjunct therapies you could team up with exosomes. So in this case, yes, it would have to be injected.
Yeah. But the second question from Dr. Mu'taz, which markers can be used for exosomes characterization, and is it make difference in the exosomes effect according to the type of the markers?
There are a number of surface markers, CD63, 9, and 81. They are the standard ones, but there are also intrinsic biogenesis markers like syntonine, and you should check for those as well, because otherwise you might actually have an extracellular vesicles with a particular surface marker and not an exosome. The same is actually true for lyophilized exosomes. If they tell you that they tested for CD63, for instance, and therefore confirm the presence of an exosome, that is not necessarily the case, because it can be a fragment of the exosome that had been destroyed that is still CD63 positive. So there are a number of biomarkers that actually can and should be used, and it's a combination of many of them. So we are testing 6 markers in this context. All
right. I think Mary's phone, she was not satisfied about the answer of the pH question. So she asked again, what is the ideal pH range of a solution that can be used to mix with Hg0 exosomes to create a stable, effective cocktail, given that the pure solution is very liquid? Additionally, what viscosity enhancers are recommended to maintain bioactivity?
What was the last? What kind of enhancers?
Yeah. Additionally, what viscosity enhancers are recommended to maintain bioactivity?
Yes. So I think there was an earlier question.
Yeah, from Dr. Mahesh about mixing with
hyaluronic acid would be one. You could mix it up with hydrogel, also very easy and of it. pH, as I mentioned, it's around the physiological environment of skin, which is 5 to 7. You want to observe, or physiological saline is oftentimes 7.4, so everything is fine. You should not go above 8 and not below 5. If it becomes too acidic, it's not good for the exosomes.
I hope this is clear, Dr. Mary. If you would like to raise your concern again, you feel free to unmute yourself and speak to Dr. Heke. If any doctor who wanted to ask or discuss his question with Dr. Heke, please feel free.
How much would you inject intralesionally into an active acne?
I would have to double check. I cannot tell you out of the top of my head how much we actually have in those case studies that I referred to earlier. So I write it down. So if you can get in touch with either Dr. Haizam or me directly, and then I'll check and give you the answer.
Thank you.
Most welcome.
This question, I think we have answered this question earlier about teen acne. There is another question from Dr. Minar. Exosomes cannot cause hyperkeratotic scars?
No, they should not.
Because it's not a question. I was just reasserting the fact that they cannot cause any hypertrophic scars, because like doctor said that exosomes actually only cause the damaged collagen to repair, not cause an overburdening of collagen.
Thank you for the support.
Yeah, that was helpful. Another question. In Taiwan, there are many companies that do exosomes IM injection, intramuscular injection. Are there any risk of using exosome to treat some diseases? What are the main reasons why FDA doesn't approve exosome for therapeutic yet? What are some risk of IV drip exosome?
That's many questions. First of all, let's start with the FDA. The FDA is always very conservative, so they're a little bit hesitant to approve anything just yet, because first of all, you have to demonstrate through years of clinical trials the efficacy and the safety profile of exosomes. So that is for anyone a long-term project, and especially the field of exosomes is still too new to actually being able to get there anytime soon. Even for stem cells, where there is now an overwhelming body of evidence that stem cell therapy is feasible and it's safe, and there's still some reluctance of the FDA to move on it. I think they are just hesitant also because they do not quite know how to govern it and how to regulate the field, and that makes it more difficult. So I think that's still a problem that will be with us for quite some time. But you can treat today under the mechanisms of right to choose. In the US, we have something that's called expanded access that allows a physician to actually treat diseases. So that's one question. What kind of diseases you can address? A lot of them. So I wouldn't rule anyone out. It's just a matter of defining how you treat it, the best route of application. We have seen exosomes to be effective, for instance, treating neurological disorders by things as simple as nasal sprays. You can also do IV, and there's no risk associated with IV drips of exosomes that I would be aware of. They have been shown to be supremely safe, always provided that they are properly manufactured and that you can trust your vendor on the clinical safety of these products. I think I addressed all questions.
Yes, correct. If there is anything remaining, I think, you replied everything about IV. Also, drip is something interesting for everyone nowadays to use exosome, but nothing yet, I think, came to United Arab Emirates as IV drip exosome.
We've also seen IV drips being applied to pain management, for instance, and there is a trend towards general rejuvenation or general well-being, like a wellness kind of approach to IV drips of exosomes that just helps the body to get stimulated wherever it's needed. So there's certainly trends out there.
Yeah. So Dr. Mary, she said, I apologize, I got logged out accidentally. So Dr. Heke replied your question, Dr. Mary, regarding the pH, that positively, yes, you can mix with something to increase the viscosity like hyaluronic or other enhancers. That pH should be between 5 to 8, right, Dr. Heke?
That's right, yeah. That sums it up.
Yeah. So the second question, some students start to interest in exosome field. Which PhD program that you would suggest them to pursue? Molecular biology or immunology or biochemistry? And if anyone who are interested in exosome, which organization or association you suggest to join?
That's a good question. It depends a little bit on where you are and what university you have in mind, and there should always be programs. In general, like with a biology major, you are on the right track, and then it depends on where you want to specialize. So biochemistry would work, but I think it's mostly molecular biology or cell biology that would be advised to engage on that route of exosome biology.
Great. There is one more question. Is there any online courses recommended for MD that you would recommend?
Unfortunately not. So what we are trying to do is we actually try to establish an oversight bureau that we hope to actually also fuel with all kinds of podcasts and webinars that are more educational, because you are absolutely right. What's missing in the field is education, where somebody is giving you hopefully unbiased take on what's possible and what is not, and to actually sort through all the noise that is in the field. Unfortunately, just like back in the days with stem cell therapy, there's a lot of black sheep in this field that have little more in mind than a quick profit, and it requires education for not only MDs but for everyone really to sort through all of this. You can always get in touch with us. I cannot provide any good platform just as of yet, but if we can help you with any kind of information that is educational, then please reach out to either me directly or to Dr. Haitham, and I will provide you with as much information as you can stomach.
The last question, Dr. Heke, was asked by one doctor that for hair restoration, is there any study show the difference between using only microneedling and microneedling with exosome? Because microneedling itself promotes hair restoration. and you are recommending to use exosome as with topical microneedling, not to inject. So is there any controlled study show the difference between using microneedling alone and microneedling with the exosome for hair restoration?
I don't think that microneedling alone is as effective, but you're right. It has been shown to actually demonstrate some kind of a benefit. I'm not sure if it's lasting, really, just like other means of hair restoration. And exosomes is a little bit different in this regard. But we are currently in the middle of executing 2 clinical trials that investigate exactly that. That actually invest microneedling versus microneedling only versus exosomes only to see what the differences are. So that's work in progress and takes a little bit longer to clearly evaluate.
All right. I think we are done. All questions were answered. Thank you so much, Dr. Heke. By the way, the webinar was recorded. I have the webinar recording, and any doctor who is interested to get the webinar, you can reach me directly or reach Dr. Heke. We will both have the webinar recorded, and feel free anytime to reach us to if you have any questions.
No problem. Thank you, Haisam, for organizing.
So thank you so much, everyone, for attending the webinar, and see you next webinar.
Thank you.
Thank you so much, Dr. Heke. Thank you so much. Thank you, Dr. Haitham.
Thank you so much. Thank you.